be successful in this class. Okay. A lot of times I wait till after exam one to do this speech because a lot of times you guys aren't listening until you get your first exam score, but I believe in you. I believe that you're listening right now. So I decided to do it now. But just kind of, there's really just three main components to it. And we can go into more detail if you want to meet with me in my office and talk about how to study, how to just kind of put your best foot forward in this class. But the kind of the basic ideas are here. So the number one thing is to show up to class ready to learn. Okay, and I've kind of already talked about this a little bit. If you show up to class and you haven't introduced yourself to this material at all yet, then a lot of it is just going to be words that you've never heard before, right? And if they're words you've never heard before, then words you've never heard before and images like figures you've never seen before, then that makes it really hard to learn the deeper concepts. And again, like I said before, like that's what I can be helpful with, is to help you understand the things that are conceptual, right? Yeah. I definitely put images on exams. Yeah, for sure. Yeah. But those are just kind of things that you can kind of teach yourself, right? The terminology, the figures. Okay, you can have your notes open while you're reading the book as well, and just say, oh, this figure's in the notes. Like I'll find that something like that in the book and make sure I understand it. Okay, so getting all that kind of like preparation for class time is the most helpful thing you can do to make the most out of class time, right? So that's number one. Number two, then when you're in class is just to be engaged, right? Like we talked about, don't have your phone on your desk, okay? Put your phone away, turn your notifications off on your iPad, since a lot of you I know are just connected with your phone and your iPad. And just be ready, be filling out the notes as we're going. Don't wait for me to uncover things if it's like something you're doing with your team, right? Because then it's like you're just copying it down. You can turn your brain off to do that, right? But if you actually like force yourself to make a decision, it's the same reason why I use the clickers, is the research shows that if you're making a decision on something, whether it's right or wrong, you learn it way better than just waiting for me to tell you what to write down, and then you write it down, right? So a lot of the notes are set up in that way that are kind of forcing you to process things during class, so you can actually learn it during class, and that makes it so you have to study less when you're not in class. And then the third thing is not about studying less, it's about studying more. And I forgot to say this on the first day, but in the syllabus, in all three sections of this class, we have a note in there that says we recommend about 8 to 12 hours per week of studying outside of class time for this class alone. And we've gotten that number by talking to students who are really successful in this class and asking them about how much time do you spend studying each week. And it's in that range. Okay. And that's a lot, right? That's a lot of time. But there's a lot of information, and it just keeps coming in this class too. And so the more, just a lot of studying every day. And then lastly is studying in an active way too. Okay. So a lot of times people will just kind of read over the notes over and over and over again, just kind of study. And then just come for the exam and just hope for the best, right? The problem is you have no idea like really what you know, right? You've just been studying everything. And the research again shows very clearly that like study and then test, study and test, study, like back and forth, that rotation of study some and then test yourself, study some and then test yourself is much more effective than just studying over and over and over again, right? You kind of get lost when you're doing it that way. So those are just a few basic tips. But again, I have more tips I can give you if you want to come to my office and talk to me at some point. Happy to give you some more guidance. But yeah, go ahead.
What was your favorite study method when you took this class?
Good question. Yeah, that's a really good question. I don't think anybody's ever asked me that question. Yeah, I think it was some version of kind of what I'm saying, like where, I mean, I did have my wife was in my study group. So me and her best friend, like the three of us studied together all the time. and she loves to tell the story that I kicked her, or I didn't want to study with them anymore when it came down to the final. I don't remember it quite that way, but but it was helpful to like study with other people, because we would talk things out and kind of quiz each other on things. But then I also just needed to like buckle down on my own a lot of times, and like, so I think it was some version of like, all right, I'm going to study this whole part of the notes and then like try to quiz myself on it in different ways. And so the big thing that I think is really good and that I did would be like with the process-based stuff, where it's like step by step by step, would be like study it, try to memorize it, and then just take a blank piece of paper and see if I can write that all out. And by doing that, put in also my own words a little bit, not just like the pure words from the notes, because that can be then it becomes harder to interpret the questions, because the questions aren't the exact words from the notes, right? So if you can kind of put it in your own words, but using the right terminology, then that can really help you, help you understand it more, right? And so that was the way to like quiz myself on that stuff. And then flashcards are really good for quizzing yourself too, right? When it comes to the just terminology and rote memorization stuff, flashcards are really helpful for that too. So yeah, good question. Thanks for asking that. Yeah. Okay. Any other questions on any of that? Okay. All right. Well, yeah, we're going to dive into the material for the day then. And yeah, we're going to talk about some of the components of cells today. So we're going to start out with one clicker question here, which is related to what we were just talking about, relating to studying. So if you've done some hiking out in the backcountry before, if you've used GPS, what do you think? You can talk to each other about it, see what you think.
You don't know where to start. Yeah.
I mean, maybe. I'll tell you what I think is the correct answer. Is it not working? Oh, because I don't have it plugged in. That's funny. Didn't think it would let me do that without plugging it in. Try it again. Is it working now? I probably have to end the class. Okay, hold on one sec. Try it one more time. You might need to hit that blue button to refresh it. There we go. All right. All right, go ahead and send something in there. All right, everybody send something in. Here's what you guys are saying. So yeah, close to 2/3 of you are saying, so you can see where you are, which is what I would contend is the most helpful part about it, because I've done backpacking in the backcountry when I don't have GPS, and then other times when I do, and it's the most helpful thing. You can see the map, you can kind of see how far it is to the peak and all these, and where to start, where some landmarks are. But if you have a map and you have no idea where you are on that map, then it's not really that helpful, right? And that's the most amazing part about going hiking with GPS the first time. It's like, whoa, I actually know where I am, right? And I think this applies exactly to what we're just talking about with studying, is because if you're just studying, you never actually stop and test yourself. You just show up for the test and you're hoping for the best, right? You have no idea if you actually know anything, right? And it's also the same thing for if somebody gets stranded out in the backcountry. What's the most helpful thing for the rescuers is to know where you are, right? And so when you come to my office and ask for help, or you go to the tutor and ask for help, I have some students that come and just like, I don't know, I'm just lost like with everything. Like, I'm like, oh, you want me to go over everything? Like, that's not helpful. Like, we can't do that, right? But if you've been testing yourself along the way, then you like, all right, I got this part, like I got this, but this is the one I have trouble with, right? So like knowing where you are is such an important part of studying. And I just always think of GPS when I think of that. So, all right. Okay, let's get into cell membranes. Okay, all right. So we've talked about phospholipids just a little bit. Okay, we're going to talk about them a lot more right here because they're the key component of cell membranes. Okay, when we look at a cell membrane, we have this model called the fluid mosaic model of cell membranes, and kind of two pieces to that, right? The fluid part is the fact that these phospholipids are kind of wiggly, right? They kind of wiggle around. They're fluid-like, they move around. The cells can change shape, they can expand, they can shrink. That kind of gives it the fluid aspect. Also, things are kind of switching around, moving locations constantly. And then the other part is the mosaic part, right? That's a tile mosaic where there's all kinds of different colors and shapes plugged into this thing. That's what we got here in a cell membrane as well. All kinds of different proteins and glycoproteins and cholesterols and other components kind of plugged into this in a mosaic-like fashion. Okay, so let's talk about phospholipids. Phospholipids are the key component here, okay? And so let's just go, yeah, with the people on your team there, answer those two questions there. Which part is polar? Which part's hydrophobic? And then just send the first one in on your clicker there. Which part is polar?
Wait, which? Yeah. Yeah, the tail. Yeah. Isn't it?
All right, about 10 more seconds. Go ahead and send something in, either A or B. Okay, looks like everybody. So 100% of you are saying A there. So you're saying that the head is polar, correct? And then what'd you say for the second question? B, the tails are hydrophobic. Okay, perfect. Okay, and it's a unique type of molecule. So again, the head's polar, the tail is non-polar. Okay, and then we've got the head being hydrophilic, meaning that it likes to be close to water, it helps kind of sticks to water. And then the tails are hydrophobic, meaning that they naturally go away from water. Okay, which this molecule is different than water, where water is polar, right? This is a molecule that has a polar side and a non-polar side. So it's both, it's polar and non-polar, which is what's called amphipathic, which is just like amphibian, right? So it has a polar part and a non-polar part to it. Okay, give you a sec to finish writing that down. If you're done, you can flip to the next page and there's a clicker question there. I've got it open. You can start talking to your teammates about that. Why do cell membranes have two layers of phospholipids?
Always a good place to start.
All right, about 10 more seconds. Go ahead and send something in here. All right, looks like everybody, and again, 100% of you have the same answer, which is C. So the non-polar tails are hydrophobic and push away water. Okay, so let's look at that with this little demonstration that your textbook has something similar to this, which is, yeah, pretty much if we took water, put it in a test tube like this, okay, and then if we took some phospholipids and squirted those on top, all the phospholipids would immediately flip over like this. All of the heads would face the water, all the tails would face up to the air, okay? Because the heads are hydrophobic, or the heads are hydrophilic, tails are hydrophobic, okay? But then if we take some more water and lightly squirt it on top of the phospholipids, then look what happens. Those half of the phospholipids flip over and the heads then push themselves up towards the water that's above, right? And then the other ones are still facing the water below, right? So we just automatically create this phospholipid bilayer, right? This just naturally occurs when we put phospholipids in water because of that property of them, right? So this function allows for water on both sides of the membrane, and then again allows the structure of this being a lipid, again makes it kind of wiggly, right? And so it allows for this balance between fluidity and also having a solid structure that seals the rest of the cell off from the outer parts of the cell. Okay, questions about any of that so far? All right. Okay, so let's talk about some of the things we find in cell membranes, okay? Talked about the main component, which is phospholipids. Now let's talk about glycolipids, okay? Glycolipids are fats or lipids with a sugar attached instead of a phosphate group, okay? This accounts for only about 5% of the membrane. And these tend to stick out on the outside of the cell. And the function of this is one of the primary glycolipids we have sticking outside of cells, help us to function for, the function is self-recognition. Okay, so this would be like the little ID card that your cells have that immune cells are going around checking if those cells are supposed to be there or not. And there's a certain, certain pattern that those immune cells are looking for. These are called MHC, major histocompatibility complex glycolipids that are telling the immune system whether that's a self cell or whether that's an invader, right? And if it's not recognized as self, then the immune system destroys it, right? So this is a type of kind of an ID card for the cells. Okay, so that's glycolipids. We also have cholesterol in the membrane. Okay, about 20% of the membrane is cholesterol. We can see those kind of embedded in between phospholipids in different places. These kind of have the they have an effect on the stiffness of the membrane. Okay, that's the best way to put it. And so overall, they kind of decrease the mobility of the membrane and kind of stiffen it in most areas. So yeah, just affecting the overall stiffness of the membrane. Okay, we also have when cells are next to each other, we have different things in the membrane that help those cells communicate or stick to each other. Okay, so most cells are held together to create a tissue, like your skin or like the things lining your blood vessels, things lining your digestive tract, your respiratory tract. Okay, most cells are sugar coated, kind of meaning that they have these little sugars sticking off of some proteins called glycoproteins, and those are sticky. Okay, and so when cells are next to each other with glycoproteins, the glyco part is the sugar part, right? And those are kind of helping the cells stick to each other some. Okay, we also have what's called gap junctions. Okay, so certain cells, when they're right up next to each other, they literally just have holes in the membrane between those two cells. Okay, and these are, connecting the cells via proteins, okay? And these are very important for tissues that are electrically excitable, like the heart muscle cells, as well as some smooth muscle cells. When we activate the top of our hearts with an electrical signal, the top part of our left ventricle, for example, that signal can pass from cell to cell to cell through those little holes called gap junctions. So that whole ventricle can contract together instead of, we wouldn't want just the top of our ventricle to contract and not the bottom, right? So that gap junction allows for that signal to just pass from cell to cell without needing the nervous system to touch every single one of those cells, right? So it's a way to kind of transport that or synchronize that electrical activity. Okay, other things we have in cell membranes that help hold cells together. One of those is called desmosomes. Okay, so desmosomes are a lot like rivets. Okay, so if you've looked at the outside of an airplane, then you see all these little round bumps on the outside of the airplane that really function to hold those metal panels on the outside of the airplane. And what those look like is kind of, we have like this big round side on one side, and then there's like almost like a nail or like a bolt going from that. And then the other side, there's also a big round side. That's just like these two things with a rod in the middle. And those just get tightened down and that holds everything together. And that's really what desmosomes look like inside of our cells. We got the flat part inside of one cell, flat part inside of the adjacent cell, and then they're just kind of stuck together like that. And that really holds our cells together tightly. And that's definitely what we have in our skin cells, right? We can kind of twist our skin and in different places, pull on it, and the cells don't easily rip apart because they're held together by these desmosomes. We also have tight junctions between cells, and these are special proteins sticking out of the cells that help to just hold the cells together as well. And these are very important for places where we don't want to just allow anything to pass through between the cells. Okay, so this is like our cells that line our intestines. Okay, we want to be in control of what goes into our bloodstream of the stuff that we eat, right? And so we have other ways to get things through, but we don't want things just kind of slipping through. So the spaces between cells are tightly bound, and those are called tight junctions. Okay, where were we? Right there. Okay, yeah, so go ahead and take a look at these three images with the people in your team, and yeah, send in the correct order. Okay, which of these answers over here is the correct order? 1, 2, and 3 of those three options.
I think 3 is.
Yeah.
I think the first one is
Okay.
like.
Okay.
Yeah.
Uh,
Is that?
yeah.
Tell me why you think.
Yeah, you guys talk about it. That's better. I mean, I'll tell you the answer in a second, but you guys talk about it. Yeah. All right, so send in your answer here. All right, a few more people, go ahead and send something in. I think there's 3 more people. Okay. I'll leave it open for a sec. Looks like there's 2 of you that still need to send it in. So about 2/3 of you are saying C, about a quarter saying B, and a few saying A. So this first one here is kind of a giveaway because it looks like something's trying to get through and it can't. Right? So which one has the function of blocking things from getting through? Tight junctions, yeah. So we got that. And then this one here looks like more like a rivet because it has the big flat side on both sides, right? Inside of one cell, inside the next cell. And then it has these kind of things connecting those 2 big flat parts. And so that's a desmosome. And then we can see the signal passing from one cell to the next here. So that would be the gap junction. So that would be C. Okay. All right. Questions on that? Okay. All right. We can do this one real quick. I think I already stole the thunder of this question, but go for it.
But like your skin becomes less than 9.
All right. 3 more people. Go ahead and send something in here. All right. We got everybody, and almost everybody, 98% of you are saying C. They're held together by desmosomes. Absolutely. Okay. All right, so that's kind of talking about the cell membrane as well as different things we find in the cell membrane. Next, we'll talk about membrane vesicles. Okay, so membrane vesicles are a form of transport that cells use to pretty much what can happen is that phospholipid bilayer can kind of fold in a spot. And when it does that, it can kind of wrap around whatever it was around. Okay, this can be something coming from the outside or something coming from the inside. But that phospholipid bilayer can just kind of wrap around it to make a little mini cell, which is a vesicle that contains whatever we're trying to transport there. Okay, so the two broad types of that are exocytosis. Okay, when we're trying to release something out of the cell. Okay, so this could be things we need to get rid of, like waste or mucus, neurotransmitters or hormones that are released by those cells. Okay. And those are going to be kind of stimulated to be released by some kind of a signal to the membrane. Okay. So that's releasing things. Okay. And when we do that, we form a cell vesicle somewhere else, maybe like in our Golgi apparatus, for example, if we're making a protein. And then that little vesicle is formed, it has a little phospholipid bilayer. It can make its way to the surface of the cell and then just fuse with the membrane. And then that just becomes part of the membrane. But everything that was on the inside is now on the outside, right? Because it just kind of fused with the membrane. Okay. The opposite is endocytosis, when we're taking things into the cell from the outside. And there's 3 types. So number 1 is called phagocytosis. So this is when the cell, pretty much just part of its membrane, just kind of folds in and it just engulfs whatever is in that space. Okay. Macrophages are the big ones that do this, but there's definitely other phagocytic cells as well. Okay, so it's engulfing usually one large particle or one large little chemical or molecule of some sort. Okay, and then pinocytosis is when the cell just kind of takes a little gulp of whatever fluid is on the outside of it. It's kind of, again, folds in around it, but instead of it being like one big particle, it just takes all the fluid and everything that was in that fluid and takes it into the cell. This is the way that our intestinal cells can work at times to pull some of those food components in. And then lastly, we have receptor-mediated endocytosis. That's just supposed to be an arrow again. This is where there's some kind of receptor that's bound to by the chemical, and that tells the cell to then go ahead and ingest it or engulf it. Okay, this is how insulin works. This is how different flu viruses work as well. Okay. All right, so we've got pictures of all of these 4 different types on the next slide. Well, the 3 different types on the next slide, 3 types of endocytosis. So go ahead, same idea, which one's which, choose the one that has the correct order there, chat with your table about it and send it in.
Oh, I see.
Hey, how's it going? I did, yeah. I'll see you then. What's going on?
Am I supposed to know if that's liquid?
Yeah, I mean, the whole outside of the cell will be liquid for sure. The purpose of these is really just to kind of get you guys to process it and think about it and talk about it. Some of the images aren't like perfectly clear,
So,
but
will something like this be on the test?
potentially. Yeah.
Are the bones in the first test, or is it just?
No, the lab and lecture are separate, yeah.
Never on like platform.
No. No. Yeah, we'll talk about the joint. Yeah, if it's not in the note packet, then it's not on the lecture exam. Okay, that's just like the way to differentiate. Yeah. There is overlap later when we get to the muscles. It's in both, but it's not, if it is in the note packet, so it's on the exam. Yeah.
For like the TBO,
Uh-huh.
what part specifically should we study? The whole
So
thing?
the PowerPoint right after this one.
Oh,
Yeah. Mhm.
Oh.
Okay, so send that in if you haven't yet. All right, so most of you, actually all of you are saying D there. Everybody's saying D. So the way you can kind of tell the difference between 1 and 2, how can you tell the difference between 1 and 2? What do you see in 2? Receptors, right? That's the difference, right? You can see that there's little receptors there, kind of sending some signal to the cell, and then that tells the cell to go ahead and engulf it. Whereas this one, it's just gulping whatever's there, right? It's just kind of taking in whatever's there. And then phagocytosis is a large particle, right? And so that's how you can see the difference there. That's taking in a little bacteria, right? And you can kind of see how that little vesicle forms, right? And if a cell is going to take in a bacteria like that, then it's going to destroy it once it gets in, right? But it doesn't want to just let that bacteria in and just run amok, right? So it takes it in and it's in like a little prison cell, right? And then it can send little enzymes into that vesicle that will degrade it and destroy it, right? Okay, let me show you a quick video on phagocytosis, which is always fun.
Yeah.
Yeah, I think so. Yeah, I know. Surprised. I think it's really magnified, obviously. So yeah, yeah. Okay. All right. So then let's go ahead and switch gears to talking about the cellular organelles. Okay, so organelles mean little organ. That's what that word means, right? So it's like inside of each of our cells, we have all these little components that are helping the function of that cell, just like the large organs in our body, right? Each cell has these little organs inside of them. Okay, so it's a little organ, all right? Usually with a special function. And each of them have different functions. Okay. And they're going to be enclosed within their own membrane as well. So organelles are always enclosed within their own membrane. Okay, and we only find organelles within what are called eukaryotic cells, right? So complex cells, when we just have single-celled organisms like bacteria, those are just very simple cells, and they don't have all these little components on the inside, right? And so large eukaryotic cells with organelles are what give cells the power to then become a multicellular organism and do all of the functions that they need to do individually inside of that cell. So the nucleus is one of those, right? So the nucleus is the site of all hereditary material. We're going to talk about DNA and stuff a little bit more on Monday. Okay, but this DNA is what's encoding for all proteins. Okay, so our genetic code is all found in the nucleus. Okay, mitochondria. The tagline of mitochondria is powerhouse of the cell. That's like the everlasting meme. It just never goes away. So yeah, the powerhouse of the cell. Okay, it produces ATP, right? We talked about ATP last time with the nucleic acids. And so we need lots of ATP all the time. Mitochondria produce 99% of that for us. Okay, it's unique as an organelle because it has both an inner membrane and an outer membrane. Okay, and that inner membrane is very folded, Those folds are called cristae. And so the reason it's so folded is to increase surface area. Okay, and the reason we need surface area is because we need to plug in a bunch of proteins on that membrane. And so if it's just one straight kind of oval shaped thing, then we would just have that one layer of membrane to plug the proteins in. But since it's folded, that just gives us a lot more space to be able to plug all those proteins in. This is where we do oxidative phosphorylation, right? The electron transport chain, other components of the oxidative system to make ATP, which we'll get into later. Okay. All right, ribosomes. So ribosomes are not an organelle themselves. They're kind of this little molecular machine that we find bound to one of our organelles, right? We find it bound to the rough ER, rough endoplasmic reticulum, which is an organelle. But these little ribosomes are, yeah, just like these little molecular machines that are kind of the little motors behind making the proteins for us. And that'll be part of what you study for the TBL day on Monday. Okay, their job is to take messenger RNA and convert that into a protein. Okay, and we're going to find that rough ER right on the outside of the nucleus, right? So this purple one is the nuclear membrane, nuclear envelope, and then right outside that is the rough endoplasmic reticulum, and all these little red dots are ribosomes. Okay, so overall, we have this kind of structure of a bunch of connected organelles, which is called the endomembrane. Okay, and that endomembrane is, again, this system of linked organelles, and its function is to produce, store, and export molecules, specifically proteins, but also fats as well, and then also helps us to degrade potentially harmful substances. Okay, so we're starting with the nucleus, going out to the rough ER, going to the Golgi apparatus, and then eventually going to a vesicle like this that can then go fuse with the membrane, like we just talked about, via exocytosis and send it out. Or it can just be incorporated into something there in the cell, right? Cell's going to make a protein that it might need to use as an enzyme or whatever. And so it might just be incorporated into the cell itself. Okay, so the rough ER, again, we were just talking about it, but this, again, closest to the nucleus. The ribosomes there are to help us produce proteins. Those proteins are going to be packaged into little vesicles, and then those vesicles are formed by endocytosis from the rough ER membrane. So kind of just like the cellular membrane, we can form vesicles right there. We can form them in any of these little mini organs as well, because those also have phospholipid bilayers as well, right? So we can form those little vesicles here. Yeah. Okay, smooth ER. We don't have ribosomes here. That's why it looks more smooth. Okay, this is kind of more of a series of tubes. Kind of looks like a type of like tube coral, if you've ever been scuba diving or snorkeling and seen stuff like that, but kind of there's some coral that looks like that. And these are just filled with enzymes that mainly help us make lipids. That's kind of one of the primary things that are made in the smooth ER are all of our lipids. Okay, so this is where all cholesterol products are made, steroid hormones. Inside of our intestinal cells, we have kind of an advanced smooth ER that's going to help us to absorb and synthesize and transport fats. Also very important for drug detoxification. And so, for example, in the liver cells, a very highly developed smooth ER because your liver is responsible for breaking down most chemicals, drugs, prescription or not, that are taken in by us. And so in muscle cells, we have a special version of this called the sarcoplasmic reticulum. And its job is to store calcium. So the sarcoplasmic reticulum's job in both cardiac muscle and skeletal muscles to store calcium ions and then to be able to release those calcium ions when it's time for those cells to contract. Okay, so all just different organelles that we find in cells. Give you a sec to finish writing that down. Okay. All right, and then we've got the Golgi apparatus. So the Golgi apparatus is where the proteins get to after they go from the rough ER, go from the rough ER into the Golgi apparatus. And this is where we kind of get finishing touches to the protein. Okay, so it might be adding or trimming some amino acids off of that. Could be adding sugars or phosphate groups. And then it's kind of functions as the post office of the cell. It's going to kind of tell that vesicle of proteins where it needs to go. Right? Is it going to stay there inside of that cell to function as some kind of a protein that the cell needs? Is it going to, so that would be like used in the cell, okay? Is it going to be exocytosed, for example, like insulin, a hormone needs to be released out into the bloodstream? That could happen as well. Or is it going to just be stored there in that vesicle for a while until the cell actually needs it, right? So the Golgi apparatus is kind of controlling that and creating those vesicles. Okay, and then the last two are called lysosomes and peroxisomes. They look very similar here because they're just little round circles or spheres with a double membrane. Okay, lysosomes are actually much bigger, but you can't necessarily tell that from this image. Lysosomes are full of degrading enzymes, kind of known as a cell demolition crew. Okay, so they come off the Golgi apparatus and they can destroy old organelles that aren't functioning properly. They can degrade the whole cell if needed through apoptosis. And then peroxisomes are kind of similar idea where they have special chemicals inside that are used to do certain processes. They come off of the rough ER instead. And one of their main jobs is detoxifying the cell. Okay. And one of the ways it does that is protecting from free radicals. Okay. So free radicals are protected. The cell gets protected from the free radicals by antioxidants. Right? So you have a lot of antioxidant enzymes inside of peroxisomes, and it's very common for free radicals to be formed during the process of making ATP. When we're making ATP or in the mitochondria, we're also releasing a bunch of free radicals. And if we don't have a way to deal with those, then the cell can get damaged. And so peroxisomes help with that process. Okay. All right, let's take a few minutes here and you guys can kind of work through this on your team. Just as a little bit of a recap, what we just talked about. What does each one look like? Kind of just a description of it, way for you to kind of remember and think about it. And then what are the primary functions of each?
Yeah. If someone has diabetes, does that mean that Golgi apparatus in their pancreas don't work? Or they just don't work for
It means the whole cell. So there's a special type of cell in the pancreas, the beta cells. And so the beta cells actually get destroyed. Yeah.
So they don't even have Golgi apparatus.
Yeah, those cells are just gone. Yeah. That's a great question. Yeah, that's a good connection.
Yeah, I would describe how it looks like. I'd say, no, it's like, I think also. It's like, Really? Oh, right, so. Like surrounding. CR is like this one's like longer and like. Which one is it? No, they look. Yeah, like the liver has a really long.
Stressing yourself out or getting excited. That's awesome. Yeah, this isn't my favorite part of the class either. Yeah, totally. I get excited when we get into actual organ systems too. Yeah.
Cause I was. First two letters are. Yeah. What did you guys say? No, that's good. They're kind of both, you know, way involved. But different ways. Wait, well, I'll find it. Yeah. Maybe with the NPR.
All right, let's go ahead and come back together here. And I did not write anything because I don't want you to erase what you wrote and then think that only what I wrote is the exact right answer. It's all in your notes there, but I'm happy to answer any questions that you might have on any of this right here. Any questions anybody wanted to ask here? Go ahead.
I mean, it's kind of like the appearance of like the Golgi apparatus versus the endoplasm. Just look like kind of skinny reasons.
Yeah. Yeah. So the question was, how do we differentiate between you're talking about the smooth endoplasmic reticulum probably,
Yeah,
or
yeah.
smooth ER versus the Golgi apparatus? You guys ready? Melissa has a good question here. Yeah, so if we look at the smooth ER, it is a little bit more like tubes that like stick up versus the Golgi apparatus, which is a little bit more like flattened, kind of like I don't know, pancake type things, like hollow pancakes kind of. So that is a little bit different. And then yeah, that's probably the best way to tell. Yeah. And the Golgi apparatus will always be like right next to the rough ER, where the smooth ER, yeah, most likely won't be right in line with it like that. You know?
It's interior.
The smooth ER. It usually is kind of like around the side, not necessarily in the same line as it. Yeah. So other questions on this? Yeah, go ahead. Sure. Yeah, so I mean that the endomembrane is the nucleus, the rough ER, the Golgi apparatus, and then those transport vesicles. So it's just kind of putting all that together. Yeah. Yeah.
I fully understand the difference between the lysosomes and peroxisome, and I've also peroxisome, it says they detoxify chemicals, but also doesn't the smooth ER do that too?
Yeah, the smoothie ER does as well. The peroxisomes are more, they detoxify different types of chemicals. Peroxisomes are more specialized for free radicals. Yeah, and the, yeah, you're saying the difference between these two, lysosome and peroxisome? Yeah, so it's just kind of what things they specialize in helping the cell with. That's the big thing. By looking at them, you can't necessarily tell. So I'm not gonna like have you tell the difference by looking at them, but more just knowing their functions is the better part. Yeah,
Okay.
yeah. Okay. Any other questions before we move on? Yeah, Paloma?
Yeah. Yeah.
Like fixing that? Yeah. Yeah, I know some of that happens in the nucleus itself and in the rough ER, but yeah, I'm not sure. Maybe if you learned that in another biology class, then I would trust it. Yeah. Yeah, for sure. I know you've had some more biology classes for sure. Yeah. But for this class, you just need to know what's here. So yeah, yeah, for sure. Okay, let's do one more clicker question here, which will help us decide for Monday's class. Okay, so again, you'll take this individual quiz and then a team quiz, and there's 10 points total there. And so you guys can decide how much of that you want to go to the individual versus the team within these boundaries. Okay, so I'll go ahead and open that up. You guys decide.
I'm just studying. My life is on
All right, so it's been decided. I like giving you the illusion of control here. It's never been a different vote ever in my career, but that's all right. At least you guys got to choose that. So, okay. All right. Have a great weekend. We'll see you guys on Monday.